to talk about basically is not the hormones themselves, or the hormones, individual hormones, like I was mentioning TSH and FSH and stuff. Let's just talk about hormones in general and how they attach to the receptors on their target organs. And this is where we have to talk a little bit about how I showed you earlier about the different structures, whether they're proteins or fat soluble, that kind of stuff. So we've got to go a little bit into that. So let's get into this. So hormones, they dissolve in the blood, and there's two forms. There's some that are bound to proteins and some that are free-floating. The best way I could explain this is we have two ways to come to schools. You could either walk to school or you could take it by way of the bus. Okay? So some hormones are attached to proteins, buses, or some are free-floating and just walking to school. If it's freezing outside, which ones do you think they're going to allow to go into the schools first? The free-floating ones, the ones that walk to school, or the ones that are on the bus? No, if it's freezing outside. You want to have them outside where they're not even covered by a bus, or you want to get the ones that pay for the bus and go inside? It's the free-floating ones, right? The ones that walk to school. You don't want them to freeze. At least the ones on the bus, they're not going to be so frozen. I mean, it'll be cold on the bus, but let's get the ones. And that's what happens first. What will happen first is the ones that get used up first are going to be the hormones that are free-floating. Whatever is left over, they're on reserve. They're on the bus. So once all the students come into the school, if they come in and they sit down and there's like 10 seats left, now they're going to go and say, okay, the people on the bus, the students on the bus, they can come inside the school and fill in those spots. Does that make sense? All right, so the free-floating ones are going to be used up first. Okay? So free-form diffused through the capillary. Now, we basically have two forms of hormones. If we break it down to two things. We can either say that they're lipid-soluble or fat-soluble, or they're water-soluble. Okay? The lipid-soluble ones can go right through the membrane, but the water-soluble ones can't. Okay? So, if I drew a donut, and this is not a trick question, does a donut have a hole?
No. Well, no.
Ah.
What?
There's no hole in there. You're calling that a hole, but that's not a hole. What if I took a pencil and I shoved it right through here? Then how would you explain where the hole is? You see what I'm saying? Okay? This is not going through the donut. This is not a hole. That's a hole. Now what if we just, and the same thing as windows, right? If you have a window over there, is there a hole in the wall? No, that's not a hole in the wall. That's a window. If I take a shotgun and put it right through the wall, that's a hole in the wall. Does that make sense? All right, same thing here. So we just extend these. We can just draw like this. We could have another one over here, another one over here, another one over here. What I'm drawing here is taking a blood vessel, the wall of the blood vessel, the endothelial cells that are making up that blood vessel wall, and we're taking it all around here. But if I cut right down the center, and then like this is a blood vessel here, and I cut it right down here, and now I lay it down. That's what I'm drawing here. These are endothelial cells, epithelial cells, right? Simple squamous epithelium. And there's little windows in there. These are called fenestra. Okay? This is the cell wall, the cell membrane. So you can't put anything in here, but it can pass through fenestra. Things can pass through there. Does that make sense? Okay? So we have what we could have fat-soluble stuff can pass through and enter the blood this way, through the membrane itself. But water-soluble things can't get through here. They repel. They can go through fenestra if they're there, but they can't get through the wall. So let's go back to a couple semesters ago, to the cell membrane. And what you've got here is this structure. And I know you guys have seen this before. The lollipop with the two sticks going on either side, right? You remember this. The phospholipid bilayer. Okay? This part here loves water. This part here hates water. So, one moment. So you could say that this is the intracellular fluid that's inside the cell. This is extracellular fluid. This is outside the cell. Okay? This could be the bloodstream. This part likes water. It's in contact with fluid. This likes water. It's contacted with the cytoplasm. This part hates water. Phospholipid. Phospho is the water, that's the hydrophilic end. So phospholipid hates it, right? You can't mix water and oil together. They separate. If we have a water-soluble structure, it kind of, let's say, will look like this. It's a water-soluble hormone. It can't pass into here because this part hates that part. That's a water-soluble thing. This hates water. So it will repel. Okay? Water-soluble things won't go through there. We have a problem, Houston. Okay? Lipid-soluble structures can pass through there, no problem. Water-soluble structures and fat-soluble structures that are huge act very similarly. They're just too big. If this is huge, it can't get through, pass through there. Likewise, fat-soluble structures and small hormones would be able to pass right through here because it could squeeze through here. That's why I put this in here color-coded. That water-soluble hormones and large hormones, they act very similarly. As opposed to fat-soluble or lipid-soluble hormones and small hormones also act similarly that way too. The green can pass right through, the red not. So we've got to work with two things. We have to see how hormones that are water soluble, how do they tell the nucleus inside here what to do? Because it can't go inside. The fat-soluble ones, well, they go inside. So this is what's going to happen here. The water-soluble molecules, their receptors are going to be embedded on the membrane itself. this will bind to this. Okay? This will change shape. Think as this as your key, and there's your keyhole. This will change shape and is connected to a few other things inside the membrane which we'll go through. A chain of events occurs, a cascade of events, a domino effect. This leads to this, leads to this, and then eventually something else goes on inside. So water-soluble molecules have its receptor on the membrane. Fat-soluble receptors. There's the hormone. Its receptors will be someplace inside the cell, whether it's in the cytoplasm or if this is going to be your nucleus, the membrane of the nucleus, the nuclear envelope, has the same setup as the phospholipid bilayer, except it's just 2 layers there. So if this fat-soluble hormone can pass through here, it will look through the cytoplasm to look for its receptor. If it's not in the cytoplasm, it can easily pass through the nucleus, and inside the nucleus, you can also have a receptor for fat soluble in there. So the receptors for fat soluble hormones are found intracellularly, whether in the cytoplasm or in the nucleus. The receptors for the water-soluble hormones will be on the membrane itself. Is that clear? All right. Again, that should be old news when you learned about the cell and stuff. But we're coming now into, they probably didn't talk about hormones, but they would have talked about anything that's going to bind to a receptor with the membrane itself. All right. So it's a fast review on that. We're going to go further now into it. All right. So, yeah, the brown form, like I said before, I don't know why I put this over here now, but again, that's dealing with the people on the bus. The free-floating ones will get used up first, and then the ones that, okay. All right, so we have this hormone-target cell interaction. A ligand is anything that binds to receptor. A hormone is a ligand. A key is a ligand. The keyhole is the receptor. So don't let the word scare you, ligand. That's all it is. It's just something that's going to bind to receptor. The binding site is the part of the receptor that the key goes into the keyhole. Right? And then, of course, you put the 2 together, you get the ligand binding site, and it's very specific. It fits like a locking key. Okay? A growth hormone cannot go to receptor of a parathyroid hormone. It just won't work. It has to fit like a locking key. All right? And that's just explaining it in picture form. Okay? So the only one that's going to be able to fit in there is the one with the pointy end, not the round or the 2 over here. All right? So there's very specific, and they fit like a locking key. Now, this thing called upregulation. This is what's going to happen here. Now, I'm just giving you numbers. Let's say we're inside of a cell. Okay? Outside this room is the bloodstream, but we're inside the cell. And we need glucose to come into the cell. Now, what's going to get glucose into the cell is insulin, a certain hormone called insulin. So, inside the cell, we don't know why there's no glucose coming inside. And I have receptors up there for insulin. And I'm just going to give you numbers. But I have 5 receptors out there for insulin. Maybe the insulin doesn't see my receptors. Maybe because there's only 5 receptors up there, it just doesn't see it well enough. So you know what I'm going to do? Because I need that glucose, that sugar. I'm going to upregulate. I'm going to make more proteins and receptors, or protein receptors, and instead of 5 up there, I'm going to put 100 up there. Now, you're basically making a bigger net. And what you're doing is, you're hoping that the insulin, if it's out there, is now able to see all those receptors and bind to it. That's upregulation. And now we have enough glucose going in. Now it's like, all right, I don't need all those receptors up there. We've got enough glucose. So we just downregulate. We bring those proteins back in. We break it up into amino acids. We use the amino acids for other proteins that the body needs and so forth. So that's upregulation and downregulation. Okay, questions about that? Straightforward, right? All right, so remember how I said I don't want you to memorize which ones are water soluble and which ones are lipid soluble, or you don't. But there's only a couple I want you to know, though. Okay, because we could go on and on about this, but you should know a couple. Receptors for catecholamines, epinephrine and norepinephrine. It's a group of substances that are called catecholamines. And peptide hormones. These two are, the receptors are found on the membrane. So these are the water-soluble or the water-soluble or the large hormones. Okay? They have a receptor that's going to be here. These hormones cannot get inside. But it has to send some kind of message inside to do something. Thyroid and steroid hormones. Now, steroid hormones are the ones that end with "-one". Not all of them, but most of them. Progesterone, estrone, which is a type of estrogen, aldosterone, cortisone. Okay, we can say basically, especially the ones that come out of the adrenal cortex. Okay, but those are the steroid hormones. Those are lipid-soluble ones. And those receptors will be found someplace inside the cell, whether in the cytoplasm or in the nucleus. Those are the ones that can pass through the plasma membrane. All right? So, again, just to reiterate, the membrane-bound receptors, all right, these are for proteins or hormones that cannot pass into the plasma membrane. So its receptors, they're the red here. They're the receptors that are found in the membrane. Okay? So they interact with ligands that can't pass through the membrane. It involves this whole cascade, and we're going to get into it. This whole cascade, what's going to be called the second messenger system. Why a second messenger? And where's the first messenger? Well, the first messenger is the hormone. We really never call it the first messenger. It's just kind of assumed that you know that. So this is the first messenger up here, the hormone. The second messenger is going to be someplace over here, which we'll talk about. It's inside the cell. It could be calcium. It could be a lot of different things. We'll get into that. I'm just giving an overview right now. But the chain of reactions that take place. And the ligands, as I said, they're water-soluble or large ligands. All right, so protein hormones, catecholamines, polypeptides, all right, these are large things. And that's basically what I was trying to draw for you, right? So its receptors on or in the membrane itself. Okay? Now, intracellular receptors. These are found either in the cytoplasm or the nucleus. And these for hormones that do pass through the plasma membrane. Okay? And these are smaller ligands or lipid soluble. Good examples are your steroid hormones. Remember, if they end with "-one", that's a steroid hormone. Okay? "-one" is O-N-E, right? Estrone or estrogen, progesterone, aldosterone, cortisone, okay? And thyroid hormones, that T3 and T4 that I talked about earlier. So these are for hormones that could pass right through that membrane, and someplace inside the cell you will have the receptors. Okay? Now, the membrane-bound ones, the ones here in red, and for all receptors for that matter, this will bind over here and can come right off it. So it binds over here, changes shape, does a whole chain of cascade events that happens, and then this hormone could come off and go on to another receptor. They don't get used up. They could use something in here called a G protein. Okay? One of the parts over here. So this is what I'm going to do. This is probably the hardest part to deal with in all of endocrinology. And it's all on this one slide. I'm going to read off this step by step, give a little, make sure you understand each line. Then I have a picture that I'm going to show you. And then I'm going to have a little analogy that I'm going to explain to you, and I'll put it towards a nightclub, so see if you can get that. And then that's why I wanted to make sure this guy could come in and fix this, but, and then I'll show you a little short 2-minute video. I'm hoping one of those will going to actually hit an interneuron and say, yes, that makes sense. All right? And it usually does. All right? So there's a lot of ways to see this. So for now, put your pencils down. You have everything here. You're going to see everything. I just want you to pay attention over here because this is probably the hardest thing. It's step by step a whole chain of events that takes place. So this is where a hormone, and I'm starting the hard one first, and then I got 2 other things I got to go through, and they're the easier ones. This is going to be a water-soluble hormone that's going to bind to a membrane-bound receptor, change shape, and it's going to do things inside here. And you'll see what I'm talking about. But one of those things in there is called a G protein. It has to activate a G protein. Other things don't use a G protein. This one does. So this is what happens. All right? And this is an example to increase CAMP, which is cyclic adenosine monophosphate. The hormone, as I told you before, is the first messenger. We don't really refer to it as the first messenger, but it's assumed that is the first messenger. It binds to the surface receptor. All right? Then this binds to a G protein. Kind of changes shape a little bit, and there's a G protein here that's going to actually bind to it because it just changed shape. The G protein is then activated. And what does that mean? What happens is there's GTP, which is guanosine triphosphate, binds to the G protein, but what was already on G protein was something called GDP. Guanosine diphosphate has 2 phosphates on it. So what happens is GTP comes up and knocks off GDP. That means now this G protein is activated. Once it's activated, it then tells something else called adenylate cyclase to get activated, which is also in the membrane. And keep in mind, if you didn't learn this before, if it ends with ASE, it's an enzyme. All right? Adenosine cyclase, adenylate cyclase. Okay? That gets activated. And when it does, it's going to spit out a lot of CAMP. We call it CAMP, cyclic AMP. And the cyclic AMP is our second messenger. That's going to be poured out inside the cell. And that's going to activate things inside the cell. So that CAMP is our second messenger. The first messenger was outside, the second messenger is inside. Everything in here is things to get that second messenger out. Once CAMP comes out, it binds to another enzyme called protein kinase. Remember, it ends with ASE, it's an enzyme. Phosphorylation occurs. And that means we're putting phosphates onto something. All right, so it occurs, and this requires ATP. It needs energy to do this. Okay? it's going to do this to certain proteins, and then it's going to activate more proteins to do whatever it needs to do. There's a message of this that needs to get inside here to tell inside the cell what to do. It has to go through these steps to do this. I'm sorry. I have to just teach this to you. The people who came up with this and discovered it are people who have no social life. Okay? And that's what happens. I'll explain that. It happens very rapidly, and I'll show you why. Okay? So let me explain to you let me do my analogy. Let's say I want to go to a nightclub. Okay? I don't know any nightclubs in Boston, so I'm going to use ones in New York City. All right? Back in the day. There's a lot of nightclubs in New York City, hundreds of them. Okay? I want to go to one particular one. Let's say that one is called Catacombs. Okay? There's a lot there, but I need to go to one. So there's a lot of receptors that's in the body. But me, as this hormone over here, I need to go to one particular one, like a lock and key. And I see that one, Catacombs. I see it over there. That's the one I want to go to. But who's standing in front of the door? This big guy. He's standing up like this. You got the bouncer there. All right? He's got a big G there, not for Gamma, but for G protein. All right? So I go in line, I get to the bouncer. Now the bouncer over there, with the big G, he allows me to go inside, so he's got to change shape. He's got to turn his body around and open the door. See? Now the only thing about this analogy I'm still working on, the hormone doesn't go inside. That's the only thing that's kind of a weird thing on here. But let's say that's the goal of what the hormone's supposed to do. So I want to go into the nightclub and see the show in there. But I want a good seat to see the show. So the G protein's turning around, opens up the door, and allows me to go inside. Okay? Now things are going to happen. All right? Things are going to happen. You have to unlock the door, let me inside, now I'm inside. Now I need to get to a good seat. So I can't just walk there because I don't know if I'm going to get a good seat or not. So I got to see the maitre d'. And it just happens, this is why I like this little analogy, it just happens that the maitre d's name is Camp. So his name's Camp. And he's going to take me to the great spot. The only thing is, I got to assure myself that I'm going to get a good shot. It's a good spot. So what do I have to do with the maitre d'? What do I have to do to make sure I get a good spot? Yeah, I'm going to duke him, right? I'm going to give him a couple bucks, right? To make sure I get a good... See, it's costing me energy to do that, money to do that, ATP to do that, you see? Kind of fit that right in, all right? And then he's going to walk me to a good spot right in the front of the stage. See, that's what's nice about that. All right? Does that make sense? All right? So that's like the goal. If you can understand that kind of... And then there's other ways... Well, here, let me show you the picture now. When you look at this picture up here, all right, here's the cell membrane, right? Here's the hormone in yellow. I need to get someplace in here. I got to do something. My goal, whatever this hormone is, my goal is to make a certain protein or make glucose come in, whatever it needs to do, because there's a lot of hormones. We're just doing generically right now. So I got to get a message from here inside. So here's the first messenger, the hormone. It binds to the receptor, and it changes shape and allows this relay area here. That's called the G protein. When this changes shape, the G protein changes shape too, and what happens is it gets activated. It knocks off GDP, allowing GTP to bind to it. Now this changes shape again, moves over, and now you got adenylate cyclase here. Because this has GTP on it, now this G protein activates adenylate cyclase. And this is going to, if it uses ATP, it's now going to make CAMP. The CAMP is then going to activate a protein kinase, become activated, and do whatever it needs to do, that hormone to do that. Does that make sense? You see there's a whole chain of events. All right? So let me show you a 2-minute video, this whole thing.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of cAMP from ATP. The cAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
Does that make sense to you guys? It's heavy. It's the heaviest stuff that we have to deal with for endocrinology. Step-by-step kind of thing. Open my analogy, the picture, the words, the video, something's going to click. All right. And this is another picture showing the same thing, step by step. You can do that on your own. Okay? Your book has something else. Now, the other thing I wanted to say, when we go back to this one over here, is this part here I kind of saved for later. What's happening here is that if there's a hormone that goes through this with a G protein, it happens very rapidly. I'll show you why. You see over here, there's a receptor. But this receptor can actually be bound to, there's only shown one G protein here, but this G protein over here, and over here, and over here, and so forth. So just pressing on one button here, one receptor here, can actually activate 5 or 10 different G proteins all at the same time. And when those 10 activate, eventually activate the adenylyl cyclase, you have many more of them. Does that make sense? So this here receptor, you've got G proteins, they can actually do a lot more of each. That's why hormones that are water soluble, that use the G protein, they happen very fast. Their effects happen very, very rapidly. Does that make sense? Can you visualize that? Okay. Now, there's other second messengers also. The one I want you to memorize is the one I just showed you with CAMP. But there's other ones there, and you should know, you should recognize that other second messengers. I'm not going to ask you to put those in order, the cascade events for these particular ones. You should know CAMP, but you should also recognize all the following second messengers except. Right? We did CAMP, there's calcium up here, there's something called DAG, there's IP3. So you should recognize those. Now, to put it into my scenario, in that analogy, how do you put that all together, how these come in here? It's this. They all use a G protein. We're just going to another nightclub that has a bouncer there. That's the same. But this time, instead of going in and seeing the maitre d', whose name is Cam, I'm seeing the manager, who's my brother. And the manager says, "Oh, you don't need to dupe me. Don't worry about it. I'll get you a good seat, but I don't need your money." So they don't use ATP. And he might take me a different way to get there. Maybe he might take me over the balcony and get a good seat. Another time, I could go in and say it's the manager of the place. He says, "Don't worry about it. You're the brother of the owner. Don't worry about it. We'll don't dupe me or anything, but we'll just go underneath the cellar and we'll come up this way." You see what I'm saying? So they're just other second messengers. There's other ways to get a good seat in that place. You don't need to use ATP or you don't need to use CAMP, but there's other ways you could do that. I'm not asking you to know those other ways. I'm just asking you to know the names of the people that'll be in there. Does that make sense? Okay? So that's what I'm saying on there. If you want to understand them, if you have, you're trying to go into biochemistry and you want to learn it, then this is the opportunity to know that if that's what your forte is going to be. You might as well grab that opportunity. Don't worry about the other mechanisms of the second messenger. I only did that just to be complete. Like here's one, it looks the same. Here's the hormone here, the G protein goes to the effector over here, but now you see there's no ATP. In this case, we're using IP3 instead of CAMP. All right? But this part here is all basically the same. Okay? Using this one too, if you want to look into that. This is using calcium as the second messenger. Okay? And this is just showing another thing too. With that. Okay? So these, I don't want you to memorize these. These are just showing you the names of the hormones over here, where they come. This part you need to know where they come from. But this is telling you which hormones actually do what I just showed you with the G protein. All right. The only ones I want you to know is peptide. Just know them as peptide hormones or the catecholamines. All right. Or the catecholamines is epinephrine and norepinephrine. Okay. And these are second messengers. You should be able to recognize the second messengers here. But again, I'm not going to ask you the events to make those second messengers activated. But you should recognize that these are second messengers. Okay? Fair enough? More like make a mnemonic. Now, what about another way? Again, we can have this hormone that's water soluble. Its membrane, or its receptor is membrane bound, and we have a G protein. But we have other ones that actually go to receptor that don't have a G protein. There's nightclubs out there that don't have a bouncer. So that's what this is. We're now going to show you other ways, and this is simpler. There's other ways that we can actually activate second messengers without a bouncer, without a G protein. We're bypassing that. You could just walk in. Does that make sense? Okay? So it's simpler. And the cascade events is we're just skipping that part. So the hormone, which is the first messenger, binds to the surface receptor, right? The inner portion of this receptor activates something called guanylate cyclase, which is like that adenylate cyclase, okay? We're just bypassing the G protein. This converts GTP to something called cyclic GMP. And this cyclic GMP is the second messenger in this case. All right? And then a cascade event happens, and whatever that particular hormone was supposed to tell that cell to do is the message got across. Okay? So it's basically the hormone binds over here, and there's no G protein, but now you got the guanylate cyclase is going to create the G, or the cyclic GMP, which is the second message. It's a shorter thing. You can just get into the nightclub without worrying about a bouncer there. Okay? And these are the hormones that do that. You should know the hormones that do that, but you don't have to worry about all the steps. Does that make sense? All right? Insulin's on there and stuff. Okay, this is the easiest to deal with. Now we're going to deal with the green one here. The green one here is lipid soluble. it can pass right through here. And its membrane, its receptor is either in the cytoplasm or in the nucleus. Again, whatever it is, it's intracellular. So let me just bypass what's up here, and I'll just put it in a picture form. All right? Again, put your pencils down. You don't have to, it's all up here. So here you've got the steroid hormone, the lipid-soluble hormone. So let me orient you to this picture. This is the cell membrane. Okay, all this is the cell membrane. Here's the cytoplasm, and here's the nucleus. And the nucleus has the nuclear envelope over here that has the same setup as the plasma membrane, the phospholipid bilayer. So if a hormone can pass right through here, that same hormone can pass through here. It's just two layers there. That's why it's called the envelope. It's just not the, think of this, but then we have another one over here. So it's two phospholipid bilayers. Okay? But if it still can pass through there. So what's going to happen is this hormone, which is steroid, lipid-soluble, can pass right through here. It's going to look in the cytoplasm for a receptor, can't find it. So then it passes into the nucleus. And it looks for its receptor. Lo and behold, it sees its receptor there. The receptor stays in an inactive form because they have these chaperone molecules that are on it. But as soon as the yellow here, the steroid hormone, binds to this, the chaperone molecules bounce off. Now you have this receptor hormone complex. And what that does, it's going to bind to a certain part of the DNA that is going to make a protein. Whatever that protein is, whatever that hormone was supposed to do to that cell, it's now going to make that protein. It's going to go through transcription, comes out here, does the translation. Do I have to go through transcription, translation with you guys again? No. I'm not going to go through that. All right, but you got to go through transcription, comes out here, does translation, makes the protein, whatever it needs to do. Okay? But because it has to look for its receptor over here, go in here, it has to look for its receptor in here, and this just goes to one receptor, there's not an amplification. So because of this whole thing, hormones that go this way happen much slower. Now, when I say slower, we're talking about milliseconds versus seconds. It's still fast, but it's not like, like minutes, hours and stuff. Okay?
Steroid hormones are not water-soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
And these are the hormones that do it this way. Again, you're looking at mainly the steroid hormones that end with "-one", right? So you have the sex steroids, so you get testosterone, progesterone, estrogen, progesterone, that kind of stuff. Aldosterone, cortisol, which is cortisone, okay? And the thyroid hormones, okay?